We have taken your previous classes like simple muscle twitch, okay, then effect of temperature, then multiple stimuli, okay. So now today's class will be the effect of two successive stimuli on simple muscle contraction of the gastrocnemic muscle, okay. So in objectives of today's class, first we will know about the absolute and refractive and relative refractive period. Then we will learn the effects of two successive stimuli on the skeletal muscle. Then the mechanism of beneficial effect. Okay. So can anyone tell what does this graph represents? This is a graph of action potential. Okay. So today we'll be knowing about the absolute refractive and the relative refractive period. So you know why there is negative the intracellularly when the cell is at resting stage. You know na at rest why it is negative inside the cell. You have learned from the previous theory classes. That part you know na everybody. Okay. So if this is the membrane potential, okay, what is membrane potential? Or what is resting membrane potential? So it is equilibrium, okay, it is maintained, okay. At rest, the potential of the membrane is maintained, okay. So that is the resting membrane potential. So we'll be giving stimulus from minus 70 millivolt, okay. So this is the membrane potential. It will gradually increase from negative to positive, and this is the time duration which is in milliseconds, okay. So what is this minus 70 millivolt? Which cell can anyone say? Resting membrane potential of which cell is minus 70 millivolt? Nerve. Yes. So this is a nerve cell, okay. So that is why we will be giving here stimulus, okay. So what does a stimulus, you know na? Okay, which produces a response, okay. So there can be many different types of stimulus, okay. So this when we will be applying a stimulus, suppose we have in our previous classes we have seen we are applying stimulus, suppose here, okay. So point of stimulus, then there will be latent period, then there will be gradual rise, okay, in the curvature. There will be contraction, then relaxation, okay. So what happens in a nerve cell is there must be certain threshold, okay, to produce a response, okay. So in this case it will be at least 10 to 15 millivolt, okay. So if it produces a response 10 to 15 millivolt, that time the stimulus will act on the particular cell, and there will be refractory period, okay. So when stimulus reaches at least 10 to 15 millivolt, that time it will spike, okay. It will gradually rise. If it is more than 10 to 15 or 10 to 15 millivolt, it will gradually rise, okay. It will give a spike from here till here. So there happens certain channels are there, we will be discussing, okay. So what happens when in depolarization? If you remember your action potential, what happens? What are the phases and what happens here? Okay, so here sodium gated channels will be opened. Mainly which channel? M gated channels will be opened, okay. So when the M gated channels will be open, sodium influx will take place, okay. The cell will get excited, okay. So there will be spike from here to here. There is a spike. See, it is gradually rising from here to here. From here it is having a spike, okay. So from here it fires, okay. So when it reaches the top of the peak, so at that time that channel will be closed, okay. Which channel will be closed? So now which channels? N-gated channels, okay. So now the N-gated channels will be closed. So now the sodium entrance will be, or the influx will be closed. It is not completely closed, but it will be closed, but it will be slower from here, okay. So now this absolute refractory period lasts from the starting of the firing point, okay, from the threshold when it spikes till 1/3, okay. So if 1/3 of the repolarization. Okay, suppose from here till here, if we consider, if we divide into it 3 equal parts, so at least here, okay, this part, so till here it is absolute refractory period. Okay, so after that what will happen now? Sodium influx has slowed and the gated channels are closed. Now what will happen? Potassium channels will open. Which channel of potassium? Edge gated channels. Okay, so this will open, this will cause what? Repolarization. Yes, but what will happen now? The channels are opened. Now what will happen? There will be efflux of the potassium ions, okay, from the cell. So the sodium ions are also, gated channels are also blocked. So it is not completely, the influx has slowed down and the potassium ions has, the gated channels are open. Potassium also started effluxing. So this will be at till 70%. Okay, till here it will be faster. Okay, from the firing point, from suppose from here till here and till drop, okay, it will be faster till the 70%. Okay, after 70% it will be the fall will be slowed down. Okay, it will be reduced. Okay, after that this is the after repolarization phase. Okay, till the this is, if this is the base, if this is the resting membrane potential of this cell, so if this is the base, okay, after here it will be after repolarization phase. Okay, and after reaching the base it will go further down. Okay, now so here this is also known as after hyperpolarization. Why? Because the channels are not completely blocked. Okay, some potassium ions will still get outside. Okay, then it will take some time, okay, to come to the resting membrane potential of this cell. Okay, suppose from here to here it is, suppose it has to be at least 4 milliseconds. Okay, so after crossing the baseline it will take at least how much time? Approximately 10 times. Okay, that of the of this time taken. Okay, so if it is 4 milliseconds, so how much? If we multiply 40, 4 multiplied by 10 is 40, so it will take approximately 40 milliseconds. Okay, so this hyperpolarization phase, okay, till it gets again its membrane potential equilibrium phase. Okay, so now again this sodium, potassium, ATPase, these pumps will get activated and this will get hyperpolarized. So to remember one thing, this point of stimulus or the threshold which you have given, this is directly proportional to the excitability. Okay, so if we apply more, then that suppose there, you know na, this threshold, there are various levels of thresholds. Suppose if I told that if it is more than 10 to 15 milliseconds, millivolt, if the this is threshold is less than 10 to 15 milliseconds, this is known as, this will be known as what? Will this produce any stimulus? Will this produce any stimulus? Because it has not reached the threshold, so this is known as subthreshold. Okay, this won't produce any stimulus, so it has to reach the threshold level. Okay, that is from the basement membrane potential to 10 to 15 millivolt. Okay, so it has to reach that level, then only it will fire. Okay, otherwise it won't. So no action potential will take place here. So there are like other stimulus also, other threshold also, like just above the threshold, just little above the threshold, and maximum you can give. Okay, so that is directly proportional to the excitability. Okay, so so now the theory part. Okay, so summation, there is summation. So here in here we will be applying two stimulus. Okay, two stimulus, and we will see whether the cell produces any response to the stimuli after giving its threshold level. Okay, the stimulus at its threshold level. So if you have read about the previous notes and the classes, okay, you will be knowing that is summation, one term known as summation. Okay, what does this mean? Any addition or multiplication of the stimulus that will give response in the tissues. Okay, this is also known as beneficial effect. Okay, so we can see this in muscular as well as in nervous tissues. Okay, so there are two forms of summation of by muscle. Okay, so the first one is summation of stimuli. So this is giving sub-threshold level multiple stimuli that will produce a response. So in here we are not going to perform this. We will be performing summation of contraction. Okay, this one. So it refers to the increase in actual magnitude of contraction resulting in multiple stimuli. So this one. Okay. So so here the theoretical part is written. Okay, you have to go through it. Okay, so this is the theory part. Okay, so this is the practical one, part one. Okay, so let me see picture of the drum, the cell, the stage. You have the picture now. You can open it and see in the lower part of the drum where the graph is made. You open it in your book if you have. Okay, so in the lower part of the drum you can see two strikers. If you can see, if you have opened the diagram. So here you can see the drum. Okay, there are two strikers. Okay, in between you can see there are two strikers. This is the revolving strikers. Okay, there are two arms of it. Okay, so with the help of this arm, what it does is we'll see the response of the muscle in the latent period. Okay, what happens when we apply the stimulus, the second stimuli at the latent period. So first stimuli we have given. Okay, first stimuli the curve will be like this, we have taken in our previous classes. So now the second stimuli, this. Okay, so we'll apply the stimulus here. Okay, there are two parts. If we divide the two parts of the latent period, okay, equally, so here no response is seen. Why? So it is due to the absolute refractory period. Okay, but we have in this experiment we will see if we divide the latent period into two halves. Okay, the first and the second half. So if we apply on the first part, okay, there will be no response. Okay, why? Because it falls in the absolute refractory period. Okay, so in the second part of the latent period, if we give any stimulus, there we will see response. Okay, why? Relative refractory period. Okay, but why? What happens in absolute? What happens in relative? So at that picture, I have shown you na. Yeah, what happens here? Which channel is open here? M. Here? Closed. So here, what will happen? The closed one will get opened. This one is open. So here, the closed one will get opened. So what will happen now? As a form, what will happen? This will give to summation. It will get more excited. So it will give further more response. See, that was the peak. Okay, now after giving the second stimuli at the second part of the latent period, what we are having is more excitation and more contraction. Okay, this is a skeletal muscle, if it was a skeletal muscle. Okay, so here there is the contraction is increased. Okay, the muscular contraction is increased as a summation of those stimuli when we are giving in the second part of the latent period. Everybody, this one is clear? Your theoretical part is, concept is clear about action potential, about refractory period. So in the first half, what is there? The first half, this falls under absolute refractory period. Okay, So absolute refractory period starts from where to where? Tell. I told na. Suppose this is the, listen carefully, okay? I am repeating again, okay? So if this is the resting membrane potential of the, this is a nerve cell, okay? So here it rests, okay? So inside it's negatively charged. So when I will be giving the stimulus here, okay? So there will be gradual increase, okay? If it reaches the threshold, that is 10 to 15. So from 10 to 15, it will fire. So it will spike. It will spike up rapidly fast. Okay. So it will reach there and then it will again come down faster. Okay. So from where the absolute refractory period starts? From the firing point. If it reaches the threshold, first of all, if it has to reach the threshold to provide a stimulus. So if it reaches there, it will fire the cell. It will get excited. So the channels will get open. Which channels? Will get open. So there will be influx of sodium ions. So it will spike rapidly above and it will reach here. So here what will happen? Not M. N-gated channels, yes. N-gated channels will get closed and this influx will be reduced. Okay, not completely blocked. Okay, so it is reduced here. Okay, so after reaching there, it will drop fast. So now it has, if this has to enter in the relative refractory period, it has to complete at least, if I told na, if I consider this as the repolarizing phase, even if this is the depolarizing phase, if I cut it into 3 halves, equal halves, so till here at least it has to complete. Okay, this is the absolute refractory period. It has to cross at least 1/3 of the repolarizing phase. Okay, so if I divide it into 3 equal parts, so this point at least till here is the absolute refractory period. So what is happening in the second half? So this is the first half. Okay, so why there is no response? This is because it falls in the absolute refractory period. That is the first half of the latent period. That is the absolute refractory period. So if we apply stimulus there, so no response will be seen. Okay, see, in the first half of the latent period, here any response you have seen? No response, no? So in second part, if we give response, so there is increased contraction in the muscle. So now you can understand. So here what happens? It goes in the relative refractory period. Okay, in the second half. Okay, so here what happens is, why there is response? Those channels which were blocked here, which channels was blocked here? N-gated channels. So this will get open. So the cell will further excite and it will produce much more response. Okay. Now it is clear to you. Okay. So this is the main thing happening. Okay. And this will be asked in our exams. Okay. So in successive stimuli, as it is entered in the relative refractory period, so there will be response. Okay, if you give response in the contracted phase, so the first response was in the first half, so there was no response. In the second half of the latent period, there was a response. Okay, so in contraction phase, it has again produced some response. So why? You know why? Then what is happening inside the muscle cell? What is the theoretical part? Why much more contraction is happening? Okay, that is one part. What is actually about the skeletal muscular part? What is the mechanism? We have shown you in the previous classes, practical classes. Why there is increase in stimulus? There was much more response. So first tell me what are the factors on which the latent period or mainly the velocity of the nerve on which we are giving the stimulus depends. So first we have to know what are the factors on which the latent period of the nerve or the sciatic nerve where we are giving the stimulus depends. There are mainly 5 factors, okay, on which the velocity of the sciatic nerve depends. The first one is there must be myelination. Okay, the first factor is whether the nerve is myelinated or not. If it is myelinated, then there will be faster conduction. Okay, what is the second point is the diameter of the nerve. Okay, and third factor is temperature. Then fourth one is at which point we are giving the stimulus. If it is towards the vertebral end, then it will take more time, and if it is close to the neuromuscular junction, it will take faster time. Okay, and what is the other point? How much stimulus we are giving. If I told that stimulus is directly proportional to excitation. Okay, if you give more stimulus, there will be more above the threshold. Okay, it has to be above the threshold. That time it will produce a response. Okay, so these are the 5 factors. Okay, and what are the factors on which the contraction and the relaxation of the muscle depends? The first one is temperature. Okay, on successive stimuli there is more increase in temperature, so it will give more contraction. Okay, then viscosity, then calcium ions. What is the mechanism? It has to get released from the sarcoplasmic reticulum, neurotransmitter. So it also depends on the neurotransmitters, mainly the name one excitatory neurotransmitter. Then what are the other factors? So this will eventually raise the metabolic activity, and all the enzymatic actions will take place. So this will give summation, okay, of the effects. So this will produce much more response in contraction. Okay, so in second stimuli, if we are giving it in the contracted phase, so how can we know, or how we are applying the second stimulus in the first phase, second phase, then in the contraction phase, then here it is the relaxation phase, and after relaxation phase, how we are doing here? That in the picture you can see the strikers. That is adjustable arms. There are 2 adjustable arms. Okay, it will depend on the distance of the arms. Okay, so if it is close, it will be close to the latent period. Okay, it is adjustable. See, if you read it, it is adjustable. Okay, so if it is close to the nearby, so it will be in the first, then second. If the distance it is gradually increased, the second stimuli also will be increased. Okay, like in the contraction phase and in the relaxation phase. So in contraction phase, what is happening, I have told. Okay, relaxation phase also there is response. Okay, we can see, and in the completion of the relaxation phase, if we apply the stimulus here, there is also a response, we can see. This is mainly due to summation. Okay, and the beneficial effect, and this is the reason of the why there is benefit. So this is the theoretical part. Okay, you have to know it. Okay, like what happens when the calcium is released from the sarcoplasmic reticulum, what is the mechanism going on. Okay, you have to read it. Okay, mainly those things. Okay, so here one point is there, this one, some increase in H plus ion concentration also. Okay, this is another point, you can add it with those points, okay, which I have said. Okay, the increase in potassium ion concentration. Okay, temperature part I have said. Okay,